Hello and welcome to this lesson on chromatography. Our plan for today is as follows. We'll begin by understanding the basic principles of chromatography. Next, we'll learn how chromatography can be used to separate mixtures. Finally, we'll outline how this method can identify substances within those mixtures. Let's get started. Chromatography is a powerful technique used to separate mixtures into their individual components, making it easier to analyze them. For example, if you have a mixture of different colored dyes and you want to find out what specific colors are present, chromatography can help by separating the dyes based on their unique properties. To understand how this works, it's best to go over the exact procedure to conduct paper chromatography. The materials needed for it are a strip of chromatography paper, a pencil, a ruler, a beaker or glass, a solvent, for example water or ethanol, a capillary tube or a fine brush, a mixture to be separated, for example ink from a felt tip pen, and finally a lid or cover for the beaker. Here's the exact procedure. First, we cut a strip of chromatography paper to fit inside the beaker. Then, using a pencil, draw a horizontal line about 1 to 2 centimeters from the bottom of the paper. This is the baseline. The reason we use pencil and not pen is that pen ink may dissolve into the solvent and interfere with the results. Using a capillary tube or fine brush, we can place a small dot of the mixture, for example, ink, onto the baseline. If we were to test multiple samples, we would need to place several dots along the line, ensuring they are spaced apart so they do not interfere with each other. Next, we pour a small amount of solvent into the beaker and place the paper into the beaker so that the bottom edge, below the baseline, is submerged in the solvent. We must make sure that solvent level is below the baseline to prevent the samples from dissolving into the solvent directly, and that the paper does not touch the sides of the beaker. Cover the beaker with a lid to minimize solvent evaporation. When we're ready, our apparatus will look like this. Now, we wait and observe. While this happens, let's outline the 2 stages in chromatography. First, we have the stationary phase, which is the part of the chromatography system that does not move. In the case of paper chromatography, the stationary phase is the paper which absorbs the substances. The paper acts as the medium through which the mixture will travel and is where the mixture we want to separate will start its journey. This is what we're observing. But look, the solvent has started to slowly travel up the paper, carrying the mixture components with it. As it moves, notice how different components of the mixture will travel at different rates and separate out. More soluble substances will move further up the paper, while less soluble ones will stay closer to the baseline. This is called the mobile phase. Once the solvent has traveled near the top of the paper, we can remove it from the beaker and leave it to dry. We also need to mark the furthest point the solvent reached on the paper, known as the solvent front, with a pencil, as this will be helpful for later analysis. We can now observe the different spots on the chromatogram. Each spot represents a different substance in the mixture. You can compare the positions of the spots to known reference materials to identify the substances. We will learn more about this in the next lesson. The principle behind paper chromatography is that different substances have different affinities for the stationary phase and the mobile phase. Substances that have a stronger attraction to the solvent and are more soluble in the solvent will travel further up the paper, while those that are more strongly attracted to the paper and are therefore less soluble in the solvent, will travel a shorter distance. Chromatography is a versatile technique with several practical applications across different fields. A key usage of it is testing purity of substances. When a pure substance is analyzed using chromatography, it will typically produce a single spot on the chromatogram. This single spot indicates that the substance is not a mixture and consists of only one component. Conversely, if a substance is impure or a mixture, it will produce multiple spots on the chromatogram, each corresponding to a different component. For example, if you use chromatography to separate the colors in black ink, you might see that the ink separates into several colored spots. For example, blue, red, yellow. This indicates that the black ink is a mixture of different pigments, with each having different solubilities and affinities for the stationary and mobile phases. It's important to note that sometimes two different substances in a mixture can behave very similarly during chromatography. This means they may have similar attractions to the mobile phase, the solvent, and the stationary phase, the paper, causing them to travel the same distance up the paper. When this happens, the two substances will appear as a single spot on the chromatogram, even though they are actually different. This can make it difficult to identify all the components in a mixture accurately because you might miss one if it hides behind another spot. To overcome this challenge, scientists often repeat the chromatography process using different solvents. Each solvent interacts with substances in different ways, so by testing the mixture in more than one solvent, it becomes easier to separate and identify all the components clearly. Using multiple solvents helps to ensure a more accurate analysis, making sure no substance in the mixture goes unnoticed. In the pharmaceutical industry, chromatography is crucial for testing the purity of drugs and medications. Ensuring the purity of pharmaceuticals is essential for safety and efficacy. A drug sample analyzed using chromatography should ideally show only one spot if it is pure. Multiple spots would indicate impurities that could affect the drug's performance or safety. Now, for some extension knowledge. Let's explore how chromatography is being used to tackle an emerging global health threat: counterfeit and substandard vaccines. These fake vaccines not only fail to protect against diseases, but can also harm individuals and undermine public trust in vaccination programs. To combat this, scientists are leveraging advanced chromatographic techniques combined with machine learning, a form of artificial intelligence, to distinguish between authentic and counterfeit vaccines. A recent breakthrough study introduced a new method that pairs chromatography with mass spectrometry, an advanced analysis tool. This technique allows researchers to examine the molecular components of a vaccine with incredible precision. By analyzing vaccines at this level, scientists can quickly and accurately determine if a vaccine is genuine or fake. This technology has the potential to transform public health efforts, ensuring that only safe and effective vaccines are distributed, and helping to restore confidence in vaccination programs worldwide. In today's lesson, we delved into the basic principles of chromatography, a key technique in chemical analysis. We learned that chromatography is a technique used to separate and identify the components of a mixture based on their solubility and attraction to different phases. We then walked through the step-by-step procedure for conducting a paper chromatography experiment, from preparing the paper and applying the sample to developing the chromatogram. We recognized that this process involves two main phases: the stationary phase, which does not move, and the mobile phase, which moves and carries the mixture along with it. Understanding chromatography allows us to delve deeper into the composition of mixtures and appreciate the intricate processes used to analyze and identify substances. Watch the rest of the GCSE chemistry course at Sophos Education.